My name is Jorge Riera. I am Associate Professor at the Department of Biomedical Engineering. I am the Director of the Neuro Mass Dynamic Lab. The Neuro Mass Dynamic Lab have been active for about 8 years. 5 of this 8 years while I was Associate Professor at the Tohoku University School of Medicine, and 3 years have been active at FIU. Effort in my lab are dedicated to understand the neuronal substrate, the microscopic neuronal substrate of different modality of brain imaging that are recorded on a microscopic level. In order to do so, we have to understand how mechanisms at this microscopic level are translated to another scale, which is a microscopic scale. And that we use the level, a multi-scale approach in which we incorporate a different, an intermediate level, which is called the mesoscopic level, and we try to represent how things are translated using this mesoscopic level that represent a voxel in your brain, about 1 by 1 millimeter cube volume. One interest in the lab is trying to understand how diseases have an impact on this mechanism, what kind of signature are represented in neuroimaging when you have a disease. Right now, the main focus is in epilepsy, having an epilepsy, but recently we got a new funding to move our laboratory to a new subject, which is migraine. So I will use example of work and technique in my lab, use it in my lab in order to motivate a student to come and approach to our group and learn new technique that may be useful for your future research. So one of the areas that we are interested is in neuroinflammatory process. So most of these diseases are linked to develop of inflammation, and are develop inflammation that are triggered probably by the existence of lesion. So we have a group that use optogenetic calcium imaging and histology mainly to study astrocytes and how astrocytes are involved in inflammatory process. So one of the main interests in this group is to understand how lesion trigger the perpetuation of seizure in epilepsy. So the main idea is to use in vivo model in which we implant optical fibers on top of the head of a mice, and with some genetic modification we can target particular astrocyte to activate or deactivate them, and that way control the neuroinflammatory process. And this work have a component in vitro that we have been trying to validate that those genetic manipulation that we will apply in vivo, but in an in vitro condition. In this case, we are using a particular optogenetic approach to using light to activate astrocyte in culture astrocyte in vitro condition. So we load astrocyte with calcium dyes, and we also transduce them with a particular channel rhodopsin channels using GFAP as a promoter, and then we stimulate the astrocyte with this. Another area of interest is microcircuit, and one of the reasons that we use this is to understand how circuit is organized in the cortex of the animal. And for that, we have a group that use EEG, intracranial recording, and histology. We have a new technique that have been patented here in the United States, or is right now in the revision. And actually, we can record high-resolution EEG channel from recording from the rat, and right now it's on the commercializing state. We have also trying to create translational approach with Miami Children's Hospital and how to use this technique. And this is what we can do as using intracranial recording high resolution local field potential. So the other area of interest is neurovascular coupling, and for that we have created, so this is how neurons communicate to the vessel, And for that, we have created a group which is using mainly fMRI, EEG recording, and histology. We target neuron astrocytes. So this work is in collaboration with the Yale University, with MRI Research Center, with Dr. Famir Haide. We are able to create map of neuronal activity associated with epileptiform activity in rats that have chronic epilepsy, understand how this network is organized. We also bring this rat here to FIU, and we record intracranial, we obtain intracranial recording from local field potential and also laser Doppler fluorometry. In collaboration with Dr. Lin, we also have been developing a technique to study hemodynamic fluctuations, spatial-temporal fluctuations, hemodynamic in the brain of rat using optical imaging. So these three groups are connected by one common thing, which is biophysical modeling. We need to understand this multi-scale level, how these are connected. So we have a modeling group that uses computer and trying to develop the biophysical model. So more information about my lab can be found in the website. Thank you very much.